Hello, my name is Craig Tin. I'm the Director of Marketing at ForteBio. Welcome to our webinar. We have some interesting applications of ForteBio BLI technology to share today, courtesy of the Aptamer group, using aptamers for small molecule detection. I'll just give a very brief introduction to kick things off. And while your lines are muted, you can still chat with us if you have questions or submit questions via the Q&A panel in your auditorium view. For those who have been following ForteBio, you may have seen in the news recently that we've announced a new ForteBio brand. As part of our transition into molecular devices, we've clarified our mission, where we're focused on enabling biologics development by continuing to provide innovative technologies and workflow solutions. So some of the areas where we see ourselves contributing to your success is with accelerating selection and characterization, providing more data for more confident decision-making and lead selection, as well as adding scalability and process integration. Within the expanded portfolio of ForteBio, we continue to have our Octet and Pioneer label-free systems, as well as new products, the Clone Select Imager and Single Cell Printer, as well as the Clonetics Colony Picker. But one of the other things that we're bringing as part of a larger organization focused on integrated workflows is we have pre-configured automation solutions, customized integrations, and the consultative expertise to be able to deliver these systems. The technology behind all of the Octet systems and the Blitz as well all revolves around biolayer interferometry. I won't go too deep into the principles, except to very quickly show that it's a property of light. We have a fiber optic biosensor that a light is shining through. And as molecules bind to the surface of the biosensor, we see shifts in the wavelengths of the reflected light. This allows us to monitor binding in real time, which then allows us to calculate binding affinity and kinetics. So I just want to give you a very quick view of what's happening at the tip of the biosensor during an assay and what you would be seeing on screen as part of the data output. You'll be seeing some of these graphs in David's subsequent presentation. So just as a quick view, you can think of building this chain on the end of a biosensor, right? As molecules continue to add layers, you will continue to see shifts in the wavelength of the reflected light. So this is what the Octet and the Blitz systems are measuring. One of the really neat things about the BLI capture and binding workflow is the flexibility of the platform and the possibilities are really very open. And one of the interesting things in this webinar today is using aptamers, right, which give you a fast development time and specificity. You can have a great deal of customization in your Octet assays. All right, and now I'd like to hand over things to Dr. David Bunka of the Aptamer Group. He is the Chief Technology Officer there. David has over a decade of experience with aptamers, having isolated more than 300 against a wide variety of targets and authoring papers and book chapters on the subject. So we're very pleased to have him, and thank you for attending again.
Thanks, Craig, for the introduction and the insights into BLI, the technology, and how people can use it. And hopefully what I'm going to do in the remainder of the slides is expand on that technology and explain a little more about how we at Aptamer Group use the Octet platform, how we use BLI in our monitoring of interactions between the aptamers and their respective targets, and also explain how we can actually use aptamers to build on the capabilities of these systems, and show you later on in the presentation about how we can actually get the Octet to do things that you might not think it would be able to do. So I guess the first point in my presentation is actually to explain to those who don't know what an aptamer actually is. So an aptamer is essentially a synthetic oligonucleotide. It's a piece of DNA or RNA that's been selected from a very diverse library to do a particular job, and that job is usually binding to a target molecule. So essentially it's an affinity ligand that can be thought of as a replacement for an antibody, for example, in a whole range of different platforms, and I'll come on to some of those in a moment. So what you can see in the figures on this slide is a comparison of an aptamer and an antibody, just to give you an idea of the relative size differences. So you can see that an aptamer is a lot smaller than an antibody, which gives them a number of advantages, especially in assay design and detection platforms. So you can see how you might be able to get more aptamer onto a sensor surface, for example, so therefore get a bigger response from your target. What you can also see on the right-hand side there is a crystal structure of an aptamer interacting with its protein target. And you can see there the sort of interaction is very similar to the way an antibody would interact with its target. So that's not going to be the focus of my talk today. What I'm actually going to be presenting is represented in the middle of the slide there. So what you can see is an aptamer actually forming a cage around its small molecule target. So this is something that is quite unique to aptamers. Antibodies and larger, more rigid molecules can't do this. This is because aptamers are very flexible and undergo a lot of conformational changes when they bind to their target, and that will be key to the rest of the presentation. So aptamers really can be seen as something that fills in the gaps left in the affinity ligand market. So they have a lot of advantages not addressed by other molecules. For example, aptamers are selected entirely in vitro. This means that we can work with almost any type of target. And as Craig mentioned at start, we've worked with aptamers against a whole variety of different molecules, everything from small molecules through proteins and peptides, all the way up to cells and whole tissues. So this is all enabled by an in vitro selection process. That process also allows us to control the binding conditions of the application. So, for example, we could generate an aptamer that works in a complex matrix that might not be amenable to antibody generation. So this is particularly important for down-the-line application developments. I'm not going to talk about that in a great deal today, but we do have other webinars on our website, and if that's of interest, we'll be happy to show those to you. The other advantages of aptamers is the fact that they are produced by solid phase synthesis. So this is a well-known, well-characterized process that's readily scaled, which means batch-to-batch variability issues with other affinity ligands like antibodies are greatly reduced when it comes to aptamer use. There's also the added benefit that aptamers can be stored dry, so there is no cold chain required in their production, storage, or transport. So this slide really highlights some of the benefits of the aptamer group. So our process is 90% automated, which means that we can handle a lot of samples in parallel. It also means that we can look at lots of different conditions in parallel and find the best approach to generating your aptamer. So this gives us a very high success rate and means that we can isolate aptamers in a much more productive manner. We also recognize within the group that aptamer selection is not a one-size-fits-all approach. So we have several base approaches, one of which I'm going to talk about today for small molecule selection, and then we tailor that process to fit the customer's needs. So I like to use this tree analogy. We can parallel process and screen lots of different conditions and find the fruit faster, the fruit obviously being your aptamer. So the other benefit of aptamers being produced by solid phase synthesis is the ease with which they can be labeled. Now, everyone's familiar with routine chemistry such as amine coupling, thiol coupling, and click chemistry, and putting these groups onto the end of an aptamer is very routine and very straightforward. This makes Aptamers highly adaptable to a whole variety of different platforms. So, for example, you may take an Aptamer on one day and file couple a gold nanoparticle to it. The next day you might want to put a fluorophore onto that same Aptamer. You can do that because of this routine chemistry. And I will come back to this routine chemistry because it also feeds into the ability to attach Aptamers to a variety of different sensor platforms, including the BLI. So the first of our polling questions here is, what area of science are you actually working in? Are you involved in drug development, bioprocessing, diagnostic developments, academia, or another area of research? Okay, and let's have a look at the results. Okay, thank you for that. So, just as a very quick outline of our selection process, how we work with our customers and how we get to the end goal. So as you can see in this slide, our process is broken into several different phases, broken up by these green arrows. So those green arrows represent stop-go points where decisions are made about the progress of the project and how we progress to the next stage. So the customer is kept in the loop at every stage of this process. What you can see at the right-hand side there is an outline of the Aptamer selection process. Again, I'm not going to go into details of this today, as this is not the focus of this seminar. What I would say is our process is slightly different from others because we involve things like a feasibility study up front to give confidence and conditions that we can then take into the Aptamer selection. So the benefits that I've talked about today really mean that Aptamers are broadly applicable to all areas of the life sciences and find uses in most commonly available applications. So lateral flows, for example, ELISAs, we have worked with those. We can use Aptamers in purification columns. I mentioned before that we can tailor binding conditions. We can also tailor release conditions, and I will give an example of that down the line. We can also do much more exciting things like biomarker discovery and development of reagents for imaging and flow cytometry, but the focus of today is going to be on biosensor development and applications using the Octet. So as Craig mentioned, a lot of biosensor applications rely on attachment of something, in this case it's an Aptamer, to the surface of a sensor, and then a readout occurs when that sensing molecule interacts with a ligand in your sample analyte. In the case of Aptamers, a lot of sensing applications rely on this conformational dynamics in the Aptamer and the changing of one conformation to the other upon binding of the target. And I'm going to talk about that in more detail as we go through the remainder of the presentation. So just as an example of the sort of systems we have in the labs here, so we have a Blitz system, which is the smaller scale instrument, which is very useful for us for samples where the material is precious or there's not a lot of it available. So the 4 microliter sample volume there is very important, very useful for us to be able to screen Aptamer interactions with rare targets. We also have a higher throughput Octet system that allows us to do much more high throughput screening. But the important point here is that the probes available through ForteBio are broadly applicable to Aptamers. So we mentioned before the chemistry for attaching Aptamers to the surface, and you can see here how they could be broadly applicable to Aptamers. You could attach any sort of Aptamer to these probes and turn a generic biosensor into a target-specific sensor. So, polling question 2. Why are you considering the use of Aptamers and or BLI as a solution in your current process? Is that through general curiosity? Do you have a current need? Or is it another application? Okay, and let's take a look at the results. Okay, so the remainder of the presentation here will talk about how we use the biolayer interferometry and use that in our screening of Aptamers to identify populations that are working and to show specificity in our aptamers, and also use them in downstream processing to identify better aptamers, individuals, and fragments. So the first thing we do, you can see here, is an interaction between an immobilized target and our, what we refer to as a naive, this is an unselected population, and you can see there very little interaction in green. This is compared to the interaction with a counter target, so this is something that the customer would not want an interaction with their aptamer, and you can see there a very low level interaction. And then that is compared with an interaction with the customer's target, and you can see there a much clearer signal, a very strong binding event. So you can see their discrimination between the target in red and the counter target in blue. So this is all done on a population level. When we see data like this, we say, right, we have selected a population of aptamers. We know that there are individuals in here that are going to perform to the customer's requirements. We then use the same system to isolate individual aptamers. So we go from a population to a monoclonal, to use the antibody term. And you can see there we get kinetics out of this, much as you would do with any other sort of interaction on this sort of system. So here we're going to talk about aptamers being used for protein purification. So in that situation, you would want your aptamer to bind to the target under one set of conditions and then release under another set of conditions. So you can use those conditions to elute from your column. And here you can see a compressed image of 50 cycles of binding, washing, and releasing from that aptamer's surface. When we overlay those traces and subtract the background, you can see that 50 traces overlay almost perfectly. So this demonstrates the reproducibility and reliability of an aptamer interaction over the 50 cycles here. But the focus of today's presentation is to talk about small molecule interactions. So these are notoriously difficult molecules for affinity ligands, largely down to the size. So with a small molecule, as one can imagine, there's not a lot of scope for interactions with that molecule. So any sites that have to be taken up with fluorophores, for example, diminish the ability to recognize that molecule. So we need a way of recognizing a small molecule without the need to modify it or attach it to a surface. And this is where aptamers come in. So for the next polling question, what methods are you using to screen your small molecule interactions? Is that BLI, SPR, ITC, NMR, MSD, MST, ELISAs, or other? Okay, and let's have a look at the results. Okay, so to come back to the question of small molecule interactions, generating aptamers against small molecules can also be challenging. Again, this comes down to the problem of immobilizing a small molecule onto a surface for selection. Those interactions can sterically hinder the aptamer interaction. So if you imagine a small molecule attached to a surface, you can see how it would be difficult for an aptamer to form a cage around that molecule. So at the Aptamer group, we've taken that process and flipped it on its head and used what we refer to as a displacement selection methodology. So in this situation, the target is kept in solution, and I'll show you how that works here. So we have found a way to attach our Aptamer library to any sort of surface. This could be a sensor surface, and I'll show you some of that in a moment. It could also be a 96-well plate, for example, in an ELISA format. We wash that surface to get rid of any aptamers that are loosely associated and not attached to the surface correctly. And then we introduce the small molecule target free in solution, so there's no immobilization of the target involved. And we rely on the interaction of the target to induce a conformational change in the aptamer, and that causes the aptamer to be displaced from the surface, and we can then collect those. And you can see in this example here, we could attach a fluorophore or a nanoparticle onto those aptamers and use that as a readout. So you can see in this slide here an example of that process working. So in this case, the aptamer is labeled with a fluorophore, so we get a fluorescent reading upon release of the aptamer. And you can see as the selections progress from round 2 at the left-hand side of this graph through to round 6, you can see the selection actually happening. So as the selections progress, we get more and more binding to the positive target in red compared to the background dilution in yellow. We can also go a step further than this, so when we introduce a negative selection target, this is something that we don't want the aptamers to interact with. You can see in the first instance on R6CS, we see a drop in the signal. This is because we've removed sequences that cross-react with those two targets. But as the selections progress, again we see the enrichment in positive target binders. We can also introduce complex matrices such as plasma to get rid of background binders to other proteins that may be inherent in your system. So what you can see here is by the end of this process at R10CS2, we've developed a population of aptamers that strongly bind to the positive target in red, but show very little background binding to the negative target in blue, and we can also do this in complex matrices such as plasma. So you can see there how you might be able to use this assay in a fluorescence-based ELISA, for example, to detect, in this case, antibiotics. So this just shows the use of that platform. So we've got here an aptamer discriminating between two targets, one in green and the counter target in red, in an ELISA-like format, and you can see that response is concentration dependent. Now, contrary to popular belief, aptamers actually do work in a wide variety of different matrices. So I've shown you already a concentration dependent binding response in a nice clean buffer, but we've also shown that in plasma as well. And you can see that although there might be a slight reduction in the signal, that response is still concentration dependent. And with the right calibration curve, you can see how you could get a fluorescence measurement there that is dose responsive, and you could therefore use that for quantitation. And we've also demonstrated this in a wide variety of other common matrices, so river water, for example, milk, urine. All of these give very slightly different responses, but you can see in each case the concentration-dependent response, so you could use this for quantification. So it's a simple assay format, but what I'm going to focus on now is converting that response and validating this on the Octet. So as I mentioned before, we use a displacement approach, and we can use this on a number of different platforms. So I've shown you already ELISA plates, but now we're going to focus on the Octet probes, because that's how we screen our system. So if you imagine this rectangle representing the surface of a biosensor probe, you can see when we load the aptamers onto that sensor surface, we get a very strong loading response curve. When we then wash that surface, we see a little bit of the aptamers coming away, and eventually that system stabilizes out. And then when we introduce the target, we get a displacement from the sensor surface and a release of the aptamer. So you can see there the increasing during loading, stabilizing during the wash, and then the displacement, you can see the aptamers coming away. Now, when we do the same thing with no target present, you can see that signal is essentially flat, and so you can see there an interaction with a very small molecule actually gives us a very big response, because what we're now measuring is, rather than the interaction of that small molecule, we're measuring the conformational change of the aptamer and the release from the surface. So we've converted a small response into a very big response using the aptamer. We actually use that response in a lot of our small molecule screening. So you can see here an example of that screening. So I've just shown you a few examples of the data here so that we're not overwhelming the screen. But what you can see is we use the same approach. We load the aptamers. In this case, it's an individual aptamer onto a separate biosensor. So this is all done high throughput on the Octet. We then wash the system and then come to release. So it's important to note that while the loading levels might be slightly different here, the response that we're looking for is not dependent on that loading. So what we're looking for is the response in terms of the elution, the displacement, and you can see that here. So the control, the unselected population, is shown there in blue for comparison, and you can see some of the aptamer sequences that are in that population don't respond to this target. They're shown in grey. But there are some very clear binding aptamers shown in red, where you can see a very rapid displacement from that surface. So these are our hits, and these would then be taken forward. Now, we've actually started going a step further than that within the aptamer group. So we want to provide the best aptamer that we can. So rather than just say, here is an individual aptamer that binds to your target, we want to provide the best molecule, and in this case, we refer to that as an optamer. Now, an optamer is essentially the minimal functional fragment, and we actually find that those fragments often outperform the aptamer, and I'll show you an example of that here. So as I've showed you previously, we have the naive population showing a very low response and the aptamer showing a much greater signal, and you can see the structure of that aptamer next to the graph there. So when we come to screen for the minimal fragments, often what you find is that we've chopped away too much of that aptamer sequence and we've destroyed its ability to bind to the target. So that would be a molecule we wouldn't be interested in. Some of these reduced aptamers also knock down the affinity for the target. So again, these would be molecules that we wouldn't take forward because they're no better than the parent aptamer. However, we also find that some of these fragments actually have an improved response because they're more stable. You've got a better responder. So we refer to that as an optamer. And you can see an example of that here, and in this case we've shown the secondary structure of the optamer as well, and you can see by comparing those two, a lot of the structural features have been removed and the core binding domain is remaining. So we then use those optamers in a binding assay, and again we use the same system here. So we load up the probes with the optamers, we wash that surface, and then we carry out a concentration-dependent release assay. So again, all of this can be carried out in parallel using the parallel processing abilities of the Octet. And what you can see here when we magnify this area and align everything for the release, you can see the naive population there having very little response, and then the aptamer showing a concentration-dependent response to the target. And what we can do on the software is flip that data over and use it in a steady-state analysis to give us a pseudo KD. So you can see here a comparison of the aptamer and optamer for the same target molecule. So you can see there the optamer is giving a better response. And when we use that steady-state fit, we get this sort of data. So you can see the optamer actually has a higher affinity by around 6 to 7-fold compared to its parent aptamer. So this is actually really good in terms of development of an assay, because you've got something that actually works better in the first instance, but also because the optamer is smaller, it's easier to produce, you get better yields, it's less expensive, but also you can see how the aptamer and optamer perform compared to each other. You can see that the optamer actually outperforms the aptamer in this situation, and that also holds true when you convert to an ELISA-like format, as you can see here. So just to finalize with a real-world example, we've done this in a number of cases now, but you can see here an example where we generated an aptamer and then an optamer against a chemotherapeutic target, and you can see the target molecule there and a metabolite at the bottom of the slide, and you can see by the BLI data above it, the aptamer is able to discriminate between the chemotherapeutic and both the negative target in red and the metabolite in blue. And we can also show that there's a concentration-dependent response to that, so we can see a nice linear fit to that, and that's importantly over the therapeutic range for this target molecule. So you can see there how we could use the aptamer on the Octet platform to monitor drug levels in real time and in a concentration-dependent manner. So for our final polling question, are you interested in running this sort of assay format presented here? Like, yes, you'd be interested in running them in-house. Yes, you'd be interested in outsourcing that data and samples to us. Or no, this is not of interest. Or other. So I'll just leave you with a summary slide to say aptamers are selected keeping the end application in mind. So we select them in the conditions in the environment and the matrix that's applicable to your end goal. They're very flexible molecules, and they can be considered as an alternative to other affinity ligands such as antibodies, and they're readily optimized and readily modified to fit them into a whole variety of platforms, and hopefully we've convinced you today about their applications in BLI and how we can use the Octet to screen aptamers and use them in a high throughput manner. On to the question and answer session, and just to remind you, you can submit your questions through the Ask a Question button on the right hand side of your screen.